Hello, everyone. Welcome to the May session of the AIRR-C seminar series. Today we have a very computational heavy speaker panel with Geir Kjetil Sandve from Norway and Thomas Konstantinovsky from Israel. So we will have, as I said, two speakers today. First about 45 minutes and then starting in 1 hour, our early careers speaker, who is Thomas today. So let me just quickly introduce Geir Kjetil Sandve to you all. So he's a professor at the computer science department at the University of Oslo. And he is working on machine learning approaches for biological sequence data. And he has a few research focuses, but one of them is working on deciphering antigen recognition on the immune receptor and immune repertoire level. He has contributed seminal contributions to the AIR field. I can just cite his machine learning ecosystem for AIR receptors and repertoires, ImmuneML, but also benchmarking of machine learning approaches, as well as he likes a lot simulations, which we have very much in common. And he's also a very dear colleague. So I'm very much looking forward to your talk. As you all have seen, probably he likes long titles. So yeah, looking forward. You can start. Yes.
Yes, and then I also hope you see my screen. Yes, so thanks for your introduction, Victor. Also, as Victor commented, instead of a title, I guess I had almost like an abstract for this talk, which I called Collectively Deciphering the Rules of Immune Receptor-Antigen Binding, deeply analyze the problem, obtain the necessary data, define standardized benchmarks, and then ensure effective method comparison. So I guess one way of I wanted it a bit long because I mean something a bit specific with it. But I also made a short title to make Victor a bit more happy, which was understanding the computational problem of predicting receptor antigen binding. And my focus here is on not really understanding the receptor antigen binding as a physical mechanism, but to understand the machine learning problem, the computational problem that we are facing, and see how we can do that as a community and what it means that we are many people. So I have a PhD in machine learning as my basis, where I worked on DNA sequence patterns. I'll come a little bit back to that. Then I did statistical genomics for many years, where I focused also a bit on reproducibility, software platform development, and epidemiology. And then the last 5-6 years, I have mainly focused on immunology and increasingly on AIR. And I think since my background is a computer scientist, I'm still a computer scientist, you'll see from my talk. And one consequence is also that my slides are still in kind of computer science style. So you will discover what I mean with that quite soon, I think. So this is the outline. It's still a bit big, but I wanted to cover the field a bit broadly, and I won't spend too much time on every topic. So I'll try to say something about like, what does it mean? What did I mean with my original title as solving this as a community? Then I'll try to go a bit into DNA, protein DNA binding, which has actually a 50-year history, much longer than ours. Then I'll try to define the computational problem we are facing and show that it's not easy even to define. Before I go into some of our contributions where I've been involved, first on understanding receptor antigen binding, and then on understanding whether AIR data can be used as a biomarker. I'll look into whether we can develop AIR methodology without simulated data at all, or whether that's, in my argument, might be necessary. And I'll round off with a plea for interoperability, reproducibility, and benchmarking. So it'll start with the first. So what do I mean by kind of like solving the problem as a community? And I had these two kind of different examples, right? So how do you optimize your bike if you're a regular person like me? Well, what you do is you go to every part, you do a little bit of improvement here and there, and it doesn't matter if you kind of perfect any part. It's most important to do the major issues, get them resolved, kind of. But how do we try to understand what the universe is built of? Well, then we go together to build particle accelerators, right? With thousands of scientists coming together, and then each scientist then try to kind of solve small, tiny, complementary pieces of that puzzle. And there's two very different ways of working. And then I think, like in computational biology, we sometimes are a bit on the bike side. We have a relatively simple computational problem that we try to solve every aspect of at the same time. While my argument is that air binding and also air as a biomarker is so complex that we need to think of it more like, how do physicists study what atoms are built up of? And then I asked a few questions here. Let me just try to move something. So what shoulders are we as a community defining and solving such complementary pieces? So when new PhD students and postdocs come in internationally, will they be able to kind of find what are the different pieces of the puzzle and focus on those? And on the other side also, what are kind of our very solid findings on the computational side that we can build on? So I'll try to come a bit back to both of these. And also a note here is that, like, I think every presentation this year have to mention something about ChatGPT and so on. And I mean, this AI wave have been here for many years now. I'm a machine learning researcher myself, but I think we always have to kind of ask ourselves whether we at any point become too eager to throw every new AI breakthrough on our problem, which is always interesting to do. But I think it's important, if we want to like really succeed in the long run, we need the patience of taking the time to truly understand the specifics of our ML problem. And when we truly understand that, then we can in a way take all the best technology into it. So, but before I go more into air and air with 1 and 2 R's, I wanted to say a bit about protein DNA binding, and that is probably less known to many people listening to this. So I think the most classic example of this is finding what is called regulatory element, which is where proteins called transcription factors bind to our genome in order to regulate the expression of genes. So this field actually started in 1977. It's very interesting to see. That is actually that field as a computational field, as actually kind of a machine learning field, started in 1977. And that first paper, at least I was able to find, had phrases like it was developed using the PLC compiler from Cornell with a reference to the compiler, and then also stating like we employed standard algorithms for sorting, which was probably quite fancy then, that they didn't just write their own sorting algorithm. So this is of course a very old field, and we can then ask ourselves, should we try to learn from that field? And is it possible to learn? Or it could be that field or others, but I think it's one of the oldest in kind of sequence pattern discovery within biology, at least. So TF binding is a very different biological context. It's a protein binding to DNA. So it's very different, and it regulates expression, right? It's a part of the regular process. But also the machine learning problem is very distinct, because I'll come back to that. Since it binds to DNA, everything is much, much simpler in terms of the binding. But still, I like this here. The farther backward you can look, the farther forward you are likely to see. So this field seems to have some characteristics and challenges that I think have some striking parallels to our field. So I joined this protein DNA binding field when it was 30 years in. So roughly 30 years after 77. At that point, there was 150 methods. They made a myriad different incompatible choices and assumptions in every combination for different parts. And mostly they didn't even explicitly spell it out. So one had to kind of go into sometimes even the code in order to see actually what assumptions they did make. When they published, then every method was superior to every other available method they compared against. There were almost no objective benchmarks, so no kind of neutral benchmarks to use. And those benchmarks that were, they often couldn't conclude anything specific because the data sets were too small to see very strong, clear differences. Also, the problem itself was a bit kind of vaguely defined. It means that there was actually probably tens of different variations of the problem that different people worked on. And it wasn't always clear which method was supposed to work as what. So, for instance, are you searching for regulatory elements in promoters or enhancers for those that are into that? Also, it was approached as a generic problem, meaning that we want to find these DNA patterns where a transcription factor binds as one methodology. But in reality, as pointed out by one called Michael Eisen, all motifs are not created equal, meaning that there are different proteins with different cellular, evolutionary histories and everything. And actually, even the machine learning nature of it was different between these proteins. And now, now 20 years later almost, with probably 150 more methods or maybe 500 more methods, they are now actually also more like specializing on different types of PDOBMs. So of course it's a longer lasting field. We have more methods, but I seem to see some patterns also in our fields where we have a lot of methods. It's a lot of different choices of assumptions being made. It's not always so clear as a reader to see what is done. There is still this tendency that every method appears to be better than everything else and has to be in order to be published. We don't have good benchmarks and so on. So I'll come back to some of these. This down here is just something from my first paper as a PhD student, where I kind of tried to look into the different components of it, which I just try to systematically do for 150 different methods. And I think it would be great if somebody would do such a kind of systematization of the mathematical models, mathematical components of the methodology we use. But at the same time, this field of protein DNA or TF binding, it established some key insights that kind of everybody could mainly rely on. So one of them was that affinity contributions by individual positions are mostly independent. What this means is that if you have an A or a C, in order for one TF to bind, it's better to have an A or a C at that position. That holds regardless of what you have at other positions. So it's just like at every individual position of a motif, you want to have the kind of the best possible nucleotide, because this is a nucleotide level. It meant that the product multinomial distribution, also known as PWM, was a very suited model. It was kind of relevant for the problem. And also what is nice is that this is a very simple statistical model. It has few parameters, so it's easy to estimate. So that allowed the field to progress quite well, and it was shown that this was much better for that field than, for instance, Hamming distance-based models that we use more in our field. Also, for instance, they kind of discovered that the best model from unlined sequence sets is NP-complete, knowing that you need heuristic algorithms. And as today, all the kind of new trendy algorithms were tried, like expectation maximization, Gibbs sampling, all of these kind of things from the 70s and 80s. And of course, and they also worked well. So it's not to say that the trend is bad. It's just like that is also part of the picture. So then I'll try to kind of define the computational problem a bit before I come into what we have done. It's one way of trying to formalizing the task is to say that when we want to learn receptor-antigen binding, we want to learn a function f that goes from x to y, where x is the sequence of the receptor, which could be now the CDR3 sequence, but you can have many different definitions here, which I'll come back to. Over to y. So x is maybe something for us like a roughly 15-dimensional categorical vector, which is the sequence of the CDR3. And y could be, for instance, the binary response if you are only interested in one particular antigen. So then it looks something like this. And it's kind of a nice starting point because it's so simple, it's so clean, and it's already a lot of complexity in trying to learn such a problem. And then we, of course, know that it's much more complex in reality. We can do something similar for the task of immune state prediction. We want to learn some function h going from a big X to y, where this X is now a matrix of something like 100,000 sequences, each of a length around 15. And then again, we can say, we could have said that y is now, of course, the same binary indicator of whether a person have a given disease, or we could then say that it's kind of every conceivable immune state we could be interested in every kind of recorded infection or recorded disease type and so on. And then even the y here could be, of course, extremely high dimensional. So then it becomes formulated like this, which is maybe the most kind of complex formulation of these very simplistic things, where you have sequences of length 15, you have 100,000 of them, and you map to a vector that is, again, the same dimension as the... No, sorry, that is a new dimension that is now the number of different immune states you're interested in. So, as I kind of alluded a bit to, even if we go to this mathematical detail that I did in the previous slide, the learning problems are still ill-defined. So if we start with this, can we predict whether a given immune receptor will recognize a given antigen, which is kind of a very nice shorthand formulation. And we can, of course, say T or B cell immune receptor, and how different is it really at the machine learning side? To be honest, I don't know really myself. And then we say, of course, can we, based on heavy or light or alpha beta chains, CDR3 or also other CDRs or other parts of the receptor, predict whether we're given T or B cell and so on. And then we can say that, of course, what we're interested in is how accurately can we do this? And we could say that it's basically not whether we can predict, but on how accurately on average we can do this. And then again, of course, it depends on what receptors we have and which antigens we have. So if you ask the question of like, how well is computational prediction working for antigen, immune receptor antigen binding, it would depend on all of these things. Type of receptor, but also kind of which of the receptors we are interested in, where some might be easier to decide on, some might be harder, and so on. And probably for some antigens, it will be harder than others. So it just tells us like how enormous this task we are facing is, and coming back to why I argued for the importance of community. We can do the same. I'll not go through all the steps, but whether an individual has a given disease based on an immune receptor repertoire, which is the question of like, might AIR in the near future be suited as a biomarker? It's not too expensive to do. So one could imagine it as a early biomarker. And again, we can then ask like, how accurately can we, based on a data set of, right, 1 million, 100,000, depending on how much blood we get, for instance, clonotypes of a given receptor type again, and representation for a set of 1,000 individuals, or maybe 1 million individuals, or 100 individuals. So, which will also influence our ability to learn a model that can again predict whether some individual, which again is from some distribution, right? Is it kind of young or old? Is it people where we already have a good picture of their risk profile, or is it much more open? And then whether you have a given disease, where it will, of course, also vary a lot based on disease, and it will also vary a lot based on where in the disease progression we are doing the measurements, and so on. Yes, so I think my aim with the first 3 points here have just been to say that as a machine learner, it's an exciting and challenging field to work in. I think there are so many research opportunities that we can only try to pick some directions that we feel are kind of undercovered, and where we think that good progress can be made. But there are an enormous number of challenges to choose between. And of course, the whole community is contributing a lot. We are, compared to the fields where I came from before, we are, I think, as a community also actually better organized in a way to kind of try to know what different people are working on, and try to cover a bit complementary. So I think we are approaching this as a community, and I think we have to make these small pieces of the puzzle and be very happy and proud whenever we can have a small piece of that. And that piece can now be a piece of the biological understanding, or it can be a piece towards the computational understanding. And the latter is what I will now focus on, which is different ways where I have been involved in trying to understand something more about the computational aspect. So I started a bit about saying this: let's throw our best AI at the problem. And I think like there is a very strong intuitive motivation today that goes something like, ChatGPT is certainly in many respects very impressive. Then learning the nature of AIR binding, it requires something with impressive capabilities. So let's try to use, for instance, large language models on AIR data. And this idea clearly has something to it also, I would say. These large language models mainly learn from unlabeled data. And we have a load of unlabeled data of AIRs. In our field, we have much, much less labeled data. Also, we have seen, and it seems at least to be the case, I think, that protein language models work well for many other protein-related tasks. So that also points that it should probably work quite well for us. But do we really understand why these large language models or protein language models really work? I tried to discuss with the language processing machine learners in my environment, and I think like, yeah, it's still, we will probably know much more about in 10 years, but I think there's a lot of why it's working and what needs to be the case in order for it to work. What about natural language could have, like, how could natural language have been different and made the current models not work well? So what is kind of, which aspects of natural language are they truly building on? So I think we try to find out, to pinpoint some of these. And one of them is, I think, is that self-supervised learning, which is what is being done here, to where you kind of mask words and predict them and so on. What I think it relies on is that since the learning task is to predict hidden words or hidden parts of the content, so you kind of learn to predict also the syntax of the text. That's basically what you want to predict, what word was there, not the meaning of it. But then the assumption is that the representations you learn in these many deep layers will now also have something in common with the compositionality of semantics, meaning that if you learn good embeddings in a way inside a language model that is able to predict syntactic elements, it might also be useful for semantics. And of course, that certainly must be the case since it works so well. If we now look for proteins in general, we know that selection, like in evolution, it will influence syntax based on the semantics. So the function of the protein is depending on the syntax, and then again, whether it functions well or not will influence which mutations will survive in the evolutionary tree. But for adaptive immune receptors, the genesis of naive receptors, at least, is driven by this nucleotide level VDJ recombination. So the syntax is kind of, we all know it starts with CAS, S, and so on, right? And these patterns here have nothing to do with the binding, which happens in the 3D protein structure. And there is a risk here that since these are so different in our case, I think there is a risk that that even though you learn to kind of predict the structure, you kind of learn to predict the VDJ recombination instead of learning to predict the components that drive function or drive binding. So our suggestion here is to exploit decades of linguistics knowledge, where I like the citation, but this is my half from 2022. So it just 2023. I think it just came out now in Nature Machine Intelligence. Nature Machine Intelligence and Nature.
Yes, exactly. Yeah.
So then I wanted to take one step back, because this is very complex with these large language models. I think it's very useful. I think we should try also to understand more about what's happening. We apply the best technology that's out there. There's enormous capacity, a lot of things that works well. But at the same time, I think it's also useful to take some steps back and look at the very simplest possible components. So is antibody antigen or immune receptor antigen binding at all driven by learnable patterns? And I think we could imagine that it works like a combination lock, right? If one number is wrong, it just doesn't bind in a way. There's maybe different lock combinations that will bind, but whenever you kind of take one of the wheels and you change it to one on the other number, it doesn't tell you anything. So the slightest change in receptor would completely revamp binding behavior. Of course, there's indication that it's clearly not the case, but then it wouldn't be possible to learn anything at all. The second thing is this overall similarity-based assumption that if you do a slight receptor change, so some amino acids only change, then probably there's only a moderate chance that the binding will change. So meaning that as you kind of have very close sequences, the binding behavior will typically be quite close. You can learn from your your close neighbors. This is something that has been the basis of machine learning, like the earliest machine learning methods, like the KNN, K nearest neighbor. You look for kind of the most similar instances. The problem with it is that it's kind of, there are some theories about like that you only learn in these small boxes at a time. So you need to have information like spread out very evenly across the whole space in order to learn. I think that is also our problem, that if this is indeed the case, then we have just sequenced a few receptors. So we can only learn in that area. The third is the pattern-based, where you would say that the binding to a given antigen is mostly determined by few positions. So these few positions are in contact with antigen, for instance. If they are present, the rest of the position doesn't matter so much. If you can do that, you can also learn, because you can learn these rules that have much fewer parameters than if you need to learn it like in a combination lock setting. So what do we know? Well, if we go directly at it, we have epitope annotated data sets. They are kind of only representing positive data typically, and they are very undersampled compared to like all the data points you could have. They are maybe, what you see is thus maybe just as much influenced by VDJ recombination or recombination probability as by its binding behavior. There's very few examples where we have like a really densely sampled region of sequence space, meaning where sequence space is kind of all these 20 to the power of 15, roughly, right? Different sequences you could have in a CDR3 if you are in a simplistic model. But we have very few cases where we have like a lot of these sequences that are very similar to each other and where we could see which of these three hypotheses seem to hold. And there's, I at least am not aware of like a lot of research that have directly tried to see which of these reality assumptions seem to be holding. It's a bit different from trying to see what the biological reality is. It's more to see which models seem to be working best. And in this protein DNA binding field, it's known that it's not positional independent, like I said in the beginning, but it was also kind of established that it's close to positional independent, close enough that that model is working well. So we're trying to look into this a bit as the simplest possible way, and taking the data from Mason et al. In Nature Biotech in 2020, I think, where we have asked, do we find small patterns that only involve a very few positions with specific amino acids that have this property that if you find that pattern, if you have this pattern at the very few positions, it will bind whatever amino acids you have at the remaining position. So can you kind of find 3 positions where you have 3 particular things, and then it doesn't matter what is in the rest of the CDR3, you almost always bind. So to the right here, we can see that we are able to find some that have a precision around 0.8, something like that, which means that given that they have this small pattern of size here, 3, 4, 5, there are many of these patterns where they actually, even after accounting, of course, for overfitting and so on, that they seem to consistently then bind to one given antigen. It's only done for one antigen, HER2. It's a fixed VJ. It's a fixed light chain. I mean, compared to the whole space of what we could have explored, it's a very, very small piece of it. But at least there we find this first indication of a clear yes answer. And we try to cover this a bit rigorously. So this is a manuscript in preparation. But we didn't really solve it, so maybe you are the ones to solve it. I'm allowed to have a small arm time. So Björn Peters and I have an open PhD position where we will actually try to use IDB data and some of the machine learning methods to try to move forward on this. So yeah. So what about biomarkers? And I think what is interesting is that AIR-based immune states prediction, where we take an immune repertoire and want to predict whether you have a disease, for instance, is really this archetypal multiple instance learning problem. So what is that? Well, it's the problem where you have bags of instances. So kind of the thing you measure comes in bags. You don't know the label of each instance like you typically do in machine learning, but only for these full bags. So that's what you see in the down right corner here, that the positive bags have some red positive instances, while the negatives are only negative instances, only blue. It only needs to have one red typically to be a positive bag. And you only see the label of the bag. You don't see that this is red and the others are blue. You only see that the whole bag is positive. There's a lot of theory in this field that we can use. One thing we have tried to, for instance, ask ourselves is that since only a few of these immune cells are presumably relevant for one immune state, right? Since the immune system is handling so many immune events, then maybe methods that work on sparse models that try to learn models and penalize them very strongly, like Lasso, maybe in some settings they could work very well for our problem. And we didn't find their yes, and that's another manuscript and in preparation. Also, deep learning is popular for a reason. As I said, many of the trendy, there's a reason for methods being trendy. Neural networks are known to have large model capacity. This is kind of the first thing you learn. They are uniform function approximators. But I think much more importantly, modern deep learning allows us to tailor architectures to a problem. So this is, for instance, one where we have a convolutional layer that explicitly tries to find these small patterns in the receptors, according to this assumption. We have attention layers that handle the multiple instance learning aspect of it, and then just a prediction layer. So this was also a paper a couple of years ago. And recently, in preparation, we are now looking to whether we can include kind of prior information at a very different level into this. So in the original multiple instance learning, you don't know anything about the instances. You just know that, for instance, this person is a celiac patient, and he probably has some immune cells that reflect that, but you don't know anything more. But maybe if we have data on gluten binding immune receptors and the patterns in gluten binding immune receptors, maybe we can kind of feed that into this network here as a prior in order to kind of guide the whole neural network in a more useful representation, more useful attention association here, but also then even better representations of the receptors. So this is also in preparation. It's my main point is kind of to show that we should really exploit our ability to dive into the details of the problem, that we know our problem well, we can work with our problem very long, and then we can really tailor the machine learning to it. So some final small points I want to make, and then I asked not whether we need simulated data, but maybe the other way around. Can we really develop AIR methodology effectively without simulated data? I think I concluded I've concluded no, that available data doesn't allow it. That's my subjective opinion, at least. And I think that databases like iReceptor, IADB, VDJDB, and so on, they are large in one way. But if you compare to the sequence space of 20 to the power 15, they are extremely tiny. So the amount of data we really have compared to what we would like to operate on is very small. Then we have this that I mentioned before, the data we looked into, where we have a dense part of sequence space. It's densely sampled through metagenesis. That has been done for at least like one antigen and one antibody to start from in a way in this Mason et al. So that is like one paper, right? So we have a bit, and then I think it's hard. I'm reviewing a lot of papers. I still find it hard to distinguish what is truly well-performing methodology from just kind of chance or selection bias and so on. It's also hard as a method developer, I think, to kind of really convince that my new method is truly better on some kind of meaningful way. And it's a truly good contribution to the community. So can we simulate an AIR binding data sets at large scale? No, we cannot really. I mean, it's way too complex. The computational time to do like complete molecular dynamics simulation at full detail wouldn't work out. But based on some simplifications with a lattice and so on, we can simulate data sets that have, I mean, binding is very different, but they still have complex emergent signals. So even though it's not the same signals, they have signals that are at least a similar level of complexity, and they are still kind of emergent. It's not what we put in. We don't know which signals are in this data set. We just put in the rules kind of. I think that can really provide insight on the difficulty of the problem, indicate whether methodologies works as hoped. And I think since there's so much data here, there's actually 1 billion synthetic labeled, binding labeled antibodies here. It can be a real powerful thing to say, at least in this reality kind of that we have control over, we can know that something is truly better. And that allows us to progress as a field and be a bit more like systematic and so on. So this was a paper just last year in Nature Computational Science. And then if you go to the AIR again, well, we in my group at least, we always come back to this CMV data set from Emerson et al, which is kind of nice because it's decent size. I mean, 600 repertoires is not enormous, but it's quite decent. We know it's possible to find something there because they did so in the original publication. And other methods have been tried out on it, so at least you have some kind of reference kind of baselines that you can compare to. And there's, of course, much more. And there's also, for instance, quite a lot of data on genome-wide RNA-seq, based on genome-wide RNA-seq instead of targeted RNA-seq. But again, I think it's still hard to truly distinguish it well. So I think one thing we can do is that we can at least currently, well, so can we simulate such AIR data, so repertoire data at large scale? I think the meal aspect we can now simulate quite well. We can really control a lot of aspects like data set sizes and so on. And also the signal properties, like how sparse, how few kind of immune cells do we want to simulate to be relevant for an immune event. Also a bit of the types of signals, how they are kind of incorporated into AIRs, how the signals kind of make up the AIR and so on, which is another manuscript in preparation, which will hopefully be out this year. So the last point is a plea for interoperability, reproducibility, and benchmarking. And I just want to make the point finally, not to moralize, but also to myself. I think like, I guess here everybody can think like, I think a lot of you here have probably tried to run several different published methods on the same data set. For instance, if you have a method you want to compare against your competitors, or you want to try out different methods on different on an analytical problem, we have done so in connection with this paper on ImmuneML in Nature Machine Intelligence 2021, for instance, where we ran some different methods, some that were included, some not. It took us a real lot of time. We had to convert the data sets, of course, to match different methods. There was library version challenges. The way to use them was very different. We had to, again, collect the result outputs and so on. So I think, like, if we have proposed this ImmuneML platform. It's very open-ended. We are happy to help out people that want to integrate with that. We have put a lot of effort into documentation and trying to make it useful. It has been presented before in many relevant settings, and I think it would be really great. I think it would make it much easier to proceed well, because if a method is available there, it can be run kind of, all methods available in that platform could be run on the same data set. They could be compared, and we would kind of have a lot more at our disposal. Still, can you today kind of make an informed choice if you have an analysis you want to do? If you have a new method, how do you objectively really compare your method so that the reviewers don't kind of suspect you of having kind of selected the data and the metrics just to make your method perform well? I don't think it's so easy today. So I just say stay tuned for an AIR machine learning competition that we are planning. And when we have that one out, it will also function as a future reference, meaning that people can take part in the competition. It will be really exciting to see how different methodologies work on the varied data sets we want to provide. But also after it has been published, it will be there for years to come, that people can refer back to as an objective benchmark for. So I think it will be really helpful for method development. And finally, I think eventually we will have to start thinking carefully and scientifically about study design and the clinical context of it. Because as AIR biomarkers come nearer to clinical use, we need to also start addressing this context of generalizability. What have we really learned? What do we learn? Do we learn like causally we can say that we learn the HLA maybe? Do we indirectly learn the age and gender and age and sex and exploit that? If so, maybe that could have been done easier by questionnaire and so on. So what is the added value of the AIR biomarker? Can study designs create challenges or make sure that you learn robust models? So again, we have a paper on bioRxiv last year by Milena Pavlovich and al that we also hope that will soon be out. It's published paper. So to conclude, I think the history of related fields like protein DNA binding that have been around for a longer time might point to some black spots in our field and some research, and some research opportunities. Both receptor binding and immune state prediction are really daunting tasks, meaning that also really interesting ML problems that you can work on for decades. That's kind of why I myself left protein DNA binding. It was not so much improvement there, but in our field, it really is. I think some of the steps to better understand this, some steps are, of course, biological, to understand the binding better, to understand the immune receptors better, to understand the structure, the folding, the docking, everything. But I think part of it's also kind of just to understand some purely computational kind of machine learning aspects of it. And we also have to make sure to kind of handle these well, like statistical insight that we can really build on. And finally, to move effectively forward as a field, I think it's really crucial that we improve interoperability, reproducibility, simulation, and benchmarking. I mean, that is something you could say in every field, but I think we should also say it in every field because it's a bit boring. It's maybe not always the most rewarded, at least in terms of when we publish it. But I think in the long run, it will really pay back to us as a field. Yeah, thank you for listening.
Thanks, Geir Kjetil. Yeah, are there questions?
Yeah, Victor, here's Lady and Kelida Miha. Hi. I have actually a question regarding the benchmarking of the methods that he mentioned. So first, a comment maybe. There are a couple of papers from my group specifically, collaboration also with you, that that address the benchmarking of the annotation, so preliminary steps of analysis of antibody repertoires before even going to the analysis itself. And yeah, we need to spend some really more efforts there as a community, as reproducible results really seem to be lacking already at the pre-processing steps rather than the analysis steps. And of course, on the analysis part and machine learning, it's very important to understand how these different methods compare and against a known ground truth. So this is the comment and the question following up. You mentioned some of your methods. Unfortunately, I didn't have time, or I haven't yet looked at the paper that you have that you had cited in your slide. Geir.
Mm-hmm.
Mm-hmm. So how can I pronounce your name rightly now? I've seen it written all the time, but
yeah, Geir is very good actually.
Geir, okay, perfect. And Pavlovich, machine learning, nature, something. Can you spend a bit more, just briefly, a couple of words of what did you benchmark there, or what was the work looking at?
Yes, so let me just, I can probably just share screen again, actually. So in this ImmuneML,
yeah,
correctly, then in a way here we have made it easy to kind of try out and so on, and we have made some data sets available. And I think what is a bit important, or what can be really useful here, is that for all the methods that are included, they can be run on data sets in the exact same format. They can be run in kind of one instruction.
Mm-hmm.
So it's kind of a really nice platform to do benchmarking of different methods. When we did
Does it
this,
have all? Mm-hmm.
Yeah.
So I'll check the platform so that we don't spend time on things I can check on my own. But does it have also the option to actually integrate more methods at the same? Okay, then I'll see that. Thank you so much.
Yes, I think we really want to have more methods there, and then it's kind of, we are hoping to do a real benchmark. We didn't do a benchmark of existing methods in the field because, yeah, as I said, we have a competition now. It's such a daunting task, but we will do it now.
Yeah, then yeah. My comment also in the chat is related to that. So in case we can do this bilateral, so we can catch up on a one-on-one basis, so that we don't take the time of the group here.
That would have been great. Yeah.
Thanks a lot for the answer.
Yes, thank you.
Any more questions?
Oh, hi, Geir.
Hi.
It's Felix. I was just wondering, in terms of all these your intellectual approach to deep learning versus antigen binding. That was great. I really like that. But in terms of community and interoperability, could you consider putting a lot of your simulated data sets into the AIR data commons so people could, access them easily and try out their.
Yes,
It could
that's
be possible. We could talk to you about it. Yeah.
Yes, that's a very good question, and I think we should. I really hope we will do that very well in a way. Yeah.
Yeah. Well, we can talk to you about that. It'd be great.
Yeah.
Hey, can you hear me? Can I ask something?
Yeah.
Yes.
Yeah. Hey. So you had a slide mentioning manuscript in preparation about. Let me make sure I understood right, that you had like a antigen antibody binding where the epitope and paratope were recognized, and you bioengineered different versions with different CTR3s, and you saw that there was no effect on the binding. This is what you meant? Can you expand a little bit about it?
Yes, I can expand a bit on that. So I'm there. I'm using the data from this Mason et al. Nature Biotech 2021. So what they did there,
actually, no.
Yeah, well, yeah, so that is what they did there was to take one particular antigen, HER2, and then they took one antibody, this trastuzumab. I'm not good at pronouncing it. And then they did mutagenesis on it, actually in kind of two rounds in a way, but they ended up with a lot of antibody sequences that are a bit similar to the starting point. And then that is kind of the data set they produced. So they did mutagenesis, and then they assessed binding based on this assay. So that is the data set we took, and then we tried to see, among these thousands of sequences that are very different in a way, do we sometimes find a set of sequences? No, sorry. Do we sometimes find that when a few amino acids are present, let's say A at position 4, Q at position 6, something like that, when this is present, then there's a lot of variety at all the other positions, but no matter what we find at the other positions, they consistently bind. And that would be a bit interesting, because then it would kind of point to that for some reason these two, they determine the structure, maybe they are in contact with the antigen, and they are kind of a bit sufficient. So you can replace the rest, and probably the structure, I mean, the structure of the antibody would change, and the physical binding would change, but they are still kind of, it's a bit robust in a way, you can say, to changes in all the other places. And the reason I am so interested in that is that if that is indeed the case, it gives us really good hopes for learning. It's very similar to what kernels do in these convolutional layers of deep learning, because then there is kind of, it's a much simpler rule to say A at position 4, Q at position 6. That is kind of a low parameter. It's like a simple rule, which also deep learning can learn. It gives kind of a very simple decision boundary in terms of machine learning, a very kind of smooth decision boundary, regular decision boundary at least, as compared to if any kind of combination of amino, because that's sometimes what I also hear, right? That if you change one amino acid, then everything of binding will be completely different. And then it's kind of a bit hopeless to do machine learning, because if you cannot interpolate or even extrapolate beyond what you have observed, then you cannot have meaningful prediction. What did that kind of clarify?
Looking forward for this paper. Okay, thank you.
Any more questions? Maybe then I have one last question, maybe quick answer. So one can do benchmarking with experimental data sets, with simulated data sets. What is preferred? Yeah.
Yes, I think one thing I see is that when people do benchmarking with experimental data sets, they very often don't include uncertainty of their estimates. So when you see that a method works with a performance of 78%, you have an estimate based on a few data points. You want it to work, of course, on more than exactly those. But they very seldomly put the confidence intervals or something like that. I think the reason is that often if you would do so, these confidence intervals will be so large that any difference you see, any signal can you see in a bar plot, anything, is just like washed out by these enormous uncertainties. That is at least a risk. That is what we saw in this protein DNA binding field. So what does it mean? It means you cannot really separate chance out of the picture. It could be a difference, something could be better, or it could be chance, and you have really no clue. With simulated data, you cannot guarantee that it's realistic in a way, that is really solving the same problem, and it has a lot of caveats. But you can know that in this simulation model, this thing, like in this unconstrained generation, you can actually say, we have so much data now that the confidence intervals are very small. We know for a fact that this method, given this assumption, is working better. So something about being explicit about assumptions, being sure about the conclusion, and then we can, as a field, move towards more and more realistic, but it's kind of a bit solid steps from something simplistic towards increasingly realistic. And like in 10 years, maybe we have these huge experimental data sets, and then we are all fine.
Great. I think we need to move on, but thank you very much again, Geir Kjetil.